Hi everyone, my name is Anna-Sara Krång and I'm a researcher in Marine Ecology and Ecotoxicology at IVL, Swedish Environmental Research Institute. In this presentation, I will talk about environmental concerns and possible ecotoxicological effects of bio-based plastics. So over the last 60 years, plastics production has increased greatly and continues to grow. The environmental concerns posed by plastics are related to the resource and energy demanding production, emission of greenhouse gases, the enormous amount of plastic waste that is generated, and the substantial fraction of plastic waste that unfortunately ends up as litter in nature, particularly in the sea. Plastics are found all over the world and may persist for a very long time with potential hazards to biota. In nature, plastics may cause physical or mechanical stress due to entanglement or strangling. Ingestion of plastics may reduce nutrient uptake due to clogging of intestines or energy cost for digestion. Plastic may also induce toxicity due to leaching of plastic additives, degradation products or production chemicals, or exposure to contaminants sorbed to plastics. Generally, reduced use of plastics along with efficient reuse and recycling are preferable. But in some cases, bio-based and biodegradable plastics can be beneficial compared to conventional fossil-based plastics, but this needs to be carefully evaluated. Biodegradability strongly depends on the environmental conditions, and plastics that degrade relatively fast under optimal conditions in the lab or in industrial compost may persist and accumulate for a very long time if they end up in the ocean, on the seabed, or buried in soils due to littering, spills, or wear during use. In nature, degradation and fragmentation of larger plastic items due to UV light, heating, and mechanical abrasion will gradually generate numerous tiny plastic particles, microplastics, that are available for uptake in biota. In the sea, most microplastics will eventually sink to the bottom, and therefore sea sediments act as sinks for microplastics, and organisms that live on the seafloor risk high microplastic exposures in polluted areas. The effect of microplastics have on organisms may in turn be influenced by the degree of degradation and fragmentation. So in order to evaluate the environmental risk of plastics, we need to investigate their degradability under natural environmental conditions, as will be presented in other modules later on this week. And we must also study the impacts that plastics may have on organisms that live in these environments. Many kinds of tests can be done to assess toxicity of materials, including acute and long-term effects on survival, growth, reproduction, and behavior of different organisms. Within this project, several partners perform ecotoxicological tests of different plastics materials that are developed as part of the project. These tests are performed on a range of organisms, including plants, microbes, algae, and invertebrates, from terrestrial, limnic, and marine environments. Some of these tests follow established standard protocols to measure toxicity. In the rest of these units, I will focus on tests that we at IVL have done on the PLA-based plastic that is designated for mulch film to cover and protect agricultural land, which is one of the targeted applications within the project. We compare the PLA-based plastic with the corresponding conventional fossil-based plastic polyethylene that is used for agriculture mulch films today. We strive to use as environmentally realistic and relevant experimental conditions as possible. Here we investigated uptake and effects of microplastic from mulch films on the marine sediment-dwelling lugworm. Lugworms are common in shallow bays around Europe. They ingest large amounts of sediment and therefore risk high microplastic exposures in polluted areas. Indeed, high concentrations of microplastics from mulch film have been measured in sediments along coast where agriculture is abundant. Lugworms are important for reworking and oxygenating the seafloor. They are also important food for fish and seabirds and may transfer plastics and chemicals to higher trophic levels. We used microplastics in the size range of natural food particles for lugworms, and these were added to the sediment of the experimental aquaria. The concentration was relatively high and represent mainly hotspot areas for microplastic pollution. We also compared pristine microplastics with those pre-treated with UV light to be able to compare whether degradation affects the toxicity of the plastics. Our results show that lugworms readily ingest microplastics, but no adverse effects were found on lugworms' biological activity or general health. Neither the PLA-based or the conventional mulch film microplastics significantly affected burrowing activity, feeding rate, weight, survival, or oxidative stress, at least not at the concentrations tested and under these specific conditions. So this is one step forward for the development of PLA-based mulch film plastics. It should, however, be stressed that negative effects may still exist at higher concentrations or longer exposure times, or for other more sensitive species, life stages or effect endpoints not yet identified. To sum up, in this unit I have pointed out environmental risk of plastics that may end up as litter in the sea or other natural environments, and also that plastics in nature will fragmentize to microplastics available for uptake in biota. I have also highlighted that it is important to thoroughly investigate the ecotoxicity of new materials before they are introduced to the market to eliminate risk of hazardous effects on organisms living in areas affected by plastics or microplastic pollution. And with that, I want to thank you so much for your attention.